Hello, testing. Thank you. This is a agricultural and gardening horticultural show focusing on plant science, soil science, and microbiology. If you, geez, I got flustered. The information is for scientific and educational purposes, and it's fact-checked. Any of the websites or papers will be put in the description below. Today is the last week on soil, the seed production course, and then next week's into the microbes. So, yeah, I missed out on the microphone there for a bit, so I'm glad I can see the microphone button now. It's a lot better. I don't have to give it until 2 minutes, which is right now. So, yeah, if you're watching this back, fast forward to the slides, which is going to be right now. It's usually what I wait and do. Jesus, just as well, there's this little thing you can see flashing on the screen. Anyway, slides. This is the last week of the seed production course. The course went for many more weeks, but the last few weeks were on specific genes of like tomatoes or watermelons or stuff like that. So let's have a go at this week 4. Last week. Very slow. Rice, self-pollinated. Very good. Okay, let's just straight into, don't know what it's talking about, but it's the first slide. Just showing the bits of a self-pollinated crop, I suppose. The bits, the carpels, the male and the female bits. As you can see, this is a monocot, meaning both are on one plant. Superior ovary. I can't even remember what they were talking about there. This is the carpel again, where you can see the male pollen grain lands on the stigma, travels down, fertilizes the ovary. Wonder why it's a superior ovary. The seed grading and seed sorting machine. Yes, it's extremely important to sort your seeds so there's no or extremely little organic matter, because that organic matter is going to have microbes or also fungi on it, which is going to start and break down, release enzymes and start and break down your seeds. And that's when you'll get it. It'll smell bad and it won't be good. So it's very important to separate the organic matter from the seeds. And this is a big, massive machine. Oh, look, here's some tools. Here's some questions. It's all to do with rice. Okay, just motion distance. You can read that if you'd like. It's just to do with rice, though. Seed multiplication. So how do you get more and more seed? Well, you get the breeder seed, and then you start growing that, and then if it's a self-pollinated crop, you just let it self-pollinate. Keep going, pick out the best ones. You might want to rogue out the worst ones if ones go really crazy high or really dwarf or something like that. And then you'll be into this section. So you get rid of all the bad ones, and then it'll just go basic seed, and you just keep it going. Roguing all the bad, keeping all the good, get your uniform, all that sort of stuff. Make sure there's no disease. And you do that. That's what I did with buckwheat. I turned 500 grams into 5 kilos in the pre-basic seed first generation. Some more sorghum questions. If you're into sorghum, they might be relevant for you. What's this slide say? To enhance the growth and productivity of seed crop under dry land and garden land conditions, seeds are soaked in 2% potassium hydro H2PO. Oh, that's phosphorus. So potassium phosphate for 16 hours with a seed to solution ratio of 1 is to 0.06, okay, and dry it back to original moisture content of 8 to 9. They dry it back. If you're going to see, it's not suggested to, if you're going to inoculate your seeds before you plant them, it's not suggested to let them re-dry or try and re-moisture them up, because you're asking for trouble. You've just gone and put bacteria or fungus. It could be beneficial, but onto those seeds, and then you're expecting that to go into its dormant stage, form the spores, and then sit there. So it's got moisture. It mightn't happen. I don't recommend that way. I reckon if you're going to do a seed soak, which is awesome, and I recommend them, you should seed soak overnight with the solution of beneficial bacteria or fungi, and then use it that day. So you pull it out, you might want to let it dry for a few hours, and then let it use it, because it'll all be clumped together and it'll be pretty hard to separate. So let it dry out and use it within 24 hours. That's from, and what the science behind that is, once it's dried, it'll just be sitting there, and the seeds will be able to be separated easily for you to pick up in your hands and broadcast. And the moisture on the outside will be low enough where the bacteria or fungi won't have a chance to germinate. It will have gone in, because the moisture will have it there. Once it's the growth stage of the bacteria will go into its lag and log. And then secondary, it's in its secondary phase, third phase before death, it starts to produce its spores. So that's usually within the first few hours, 24 hours. So by doing that, you'll be pretty right. There's some science behind it anyway. I'm explaining these a fair bit because there's not many slides on this week. Let's have a look. Actually, I'll stop it. See if anyone's arrived yet. No one's arrived. I haven't said anything. That's all right. There's some people behind just listening. As long as they're enjoying it, I don't mind. Press the thumbs up too button, please, if you like this. You might like the next few weeks. It's going to be back in the microbes again. Yeah, this is today's. So I'm already halfway through those slides. So that's why I'm elaborating a little bit for once. Pearl milk questions, if you're into that. Here's a mechanical grater. So this is a clean to clean gum grater. So this mechanical grater, what's the bottom say? Physically pure and uniform sized seeds come out of it. It's just got a spinning wheel, and there'll be little sections in it where they allow things to drop through or drop and don't drop through. But they got the exact same uniform size seed that comes out of it. So that's the same size hole that it is. When you're getting your breeder seed, there's quite a few classifications for quality, like the class of seed, variety, lot number, germination percent, physical purity percent, name of the seed producer, the date of the test, and validity particulars. You want to label it extremely well. And that's 1, 2, 3, 4, 5, 6, 7, 8. If you put that on the front of it, you'll be pretty right, even if you include half of it. Class of seed, the variety you want, the germination percent is extremely important. Go and put out 10, and if 9 come up, you've got 90% germination. Or go and put out 100, and then you can get more accurate number. So you'd weigh, you just can add 100. Then you get 67 come up. Okay, you got 67% germination percent. Physical purity percent, that'll have to be worked out with your genetics, so you'll see where it came from. So how pure is it? So is it a homozygotic or a heterozygotic, or is the genetics very, well, the homozygotic tells you it's from one genus, so it's pretty stable. But you want to know the physical purity. The name of the seed producer, okay, the date of the test. Date of the test? Yeah, I'm also put on, not the date of the test, put on the production date. So when you've harvested these seeds and sorted them, so they're ready for storage. So after the final stage of the seeds are done, that's what I put on my dates. So for instance, if they're finished today, I'd be putting on it May 2025. Or if it's a very fine seed, like tobacco you currently do, but what else is fine? What's that? Clover seed. You might want to put a day on it because it might have only a 12-month shelf life, so the day might be more particular. But with bigger seeds like sunflowers, you just put the months down. And the validity particulars, okay, good. Explains that bit. Pollination. This was done in the first one. In the middle, the first generation progeny of a cross between two genetically different, identical inbreed lines. The individual lines are known as inbreeds. The first generation progeny cross between two identical individuals. So a cross between two genetically different an inbred is when you cross it with the same recurring from the seed producer. You inbreed them. Pure line theory, Johansson's pure line theory is that one. That's how you can do your test crosses too with your homozygotic pure ones. Breeding systems, all right. The hybrid rice production, mainly two types of breeding systems. Oh, Jesus. So I waste all our words. Three-line breeding system, otherwise known as cytoplasmic genetic male sterile. And the second one is two-line breeding system. In this system, the male sterility occurs depending on the photoperiod and temperature that prevails. So ways you can adjust your genetics. Breeding systems. In hybrid rice production, that's about breeding hybrid rice. If you're into breeding hybrid rice, definitely read that, because there's ways where you can fiddle with your genetics and get better outcomes from selective breeding. Seed treatments. Wow. See the slides. Anyway, this was one of the universities. This is an example of one of the unis I've studied at where all the slides aren't in order. There's a few different ones like that. They put out good material, but they're just not organised. And even when they run their classes, then it's not organised. But lucky for you, you don't have to sit the class, do you? You just get all the info. So I've kind of gone through that before. I remember about the 16 hours overnight seed soak. Yeah, and why would you actually do a seed soak? Well, if you're going to, at the start of your grow season, you might have just went through a bad Pythium problem last season, which is still going to sit there from all the moisture in the ground. So you might inoculate with Pseudomonas fluorescens, and then everything will be good. That's one example. And I could go on with many examples why you want to use different kinds of bacteria and fungus for your advantage. Protozoa too. Seed treatment. Oh, there are a few questions, but no. Briefly explain the seed treatment for rice. Seed soak treatment. That's one question. Another question there for rice. Let's just say more rice stuff. What are the advantages of gibberellic acid application in hybrid seed rice? Well, gibberellic acid, and when you're using gibberellic acid, there's about 100 different types of the phytohormone. You should try and get gibberellic acid number 3. It's one of the most studied and that seem to be performing of the best. So why should you use gibberellic acid for hybrid seed production? Well, gibberellic acid adjusts the plant height of the seed and pollen parents and enhances the panicle and stigma exertion. So it stretches cells. That's what it does. So if you've got a dominant DWF gene, for instance, this is something that actually works, and I've done it with it. In other words, the plant was stunted very badly. The nodes were very tight together. So you give it a course of this, all of a sudden it starts to elongate. Don't give it too much. Well, anyway, you'll work out if you give it too much, because I've done that. You'll just see it. It'll just, because it's hard to work out with purity to know its nano amount that it needs. So, yeah, start extreme, a couple of granules. If you've got your lab quality, like it's above 90%, you'll look out granules under the microphone glass. Put that in a little tiny bit and mix it onto the plant. But that's what it does, gibberellic acid. Actually, also, there's a saying there, it helps. No, it doesn't say that. It helps with seed germination. So if you were to soak your seeds in gibberellic acid, the second or third stage in, remember from when a seed germinates, it needs three things: the water, temperature, and oxygen. And then it's imbibed. What's the first thing that it does? It starts to break down. Oh, I can't remember. Anyway, there's two phytohormones involved in the process. I was going to give you a signaling cascade pathway, but I like to tell the actual facts, so I'm not going to try and make up something that it mightn't be. I can't picture it at the moment, so I'm not going to tell you. Ethylene and gibberellic acid are two vital hormones used for seed germination inside of the seed. So it's another advantage of it. See, I said inside of the seed. So by you putting gibberellic acid on the outside, that's not going to really help on the germination, because germination is when the first bit of the radical comes out. So you might want to, how would you do that? Where you imbibe, if you've got a dormancy situation, you crack the seed with the same bit of pliers, and then you'll see air. That's where you'll put in gibberellic acid. Or you might get a syringe, and through the outside of the, through the side of the tester where it's going to open up, through the side of the seed coat, you might put some gibberellic acid in that way. So that's how you get it inside of it to aid in germination. But this way aids in cell elongation and stretches the plant, which it blimmin' does. What's this site? Slay. Hybrid grain sorghum production. 3 parent lines. Wow, 3 parents. Isn't that amazing? Aliens. In this system, 3 inbreds lines. Oh, that's right. They got the R. The R is involved in different stages of seed manipulation. And it's like a checker line or something like that. Can't remember how they do it. That's the end of the slides. Holy moly. We went into a lot more detail than that. How many minutes is there? 20. Fair income. Hey, the Sunshine RB. How are you going, Sunshine RB? You're going to like next week's show, Sunshine RB, and that's on microbes in agriculture. So to finish off this course, see, there's a few more weeks, but they're all to do with their different, like this week's on corn or maize. This is maize. Seed production of maize. Oh yeah, we were saying about some charts before. Foundation certified pure seed 98 germination % minimum. There you go. They want a minimum of 90%. That's what they're trying to achieve. So if they put out, oh, you know how to work out percents for vapor-proof container. For vapor-proof container. So inside of their storage container, they got 8. And inside of their bag, they possibly got 12. Just have it under 10. Don't have problems. Hybrid seed. It's Duncan layer. I don't even know what that is. Micropile. Look at this. It's even spelt wrong. It's MICRO. That's a little bit. This is a monocot seed. It's a lot different from a dichotomous seed. Here's another seed treatment for seed soaking of your maize. Look at their nice maize. They got red and blue like I have. So they do it again the same way it was suggested earlier. Actually, it's exactly the same words. 16 in potassium dihydrogen phosphate. But you soak it in nano material, soak it in beneficials. Get yourself a packet of seed beneficials and soak it in that. And its a dilution ratio is very, very low. So you might only put in 1 gram for, say, a few ounces or a few 100 grams of water, and that's going to put all your seeds. So it'll be very concentrate. You'll get a stack on the outside. So when the seeds germinate, bang, they can get those microbe interactions. So what you'll do is you'll plant half with it and half without it, just for a test, just to prove if those microbes that you've got actually work in your environment. Because you might get a situation where they're not strong enough, or they're not in their favourable conditions, or they weren't stored correctly, or the colony forming units is too low. So there's many different factors that can happen. That'll be that bit. Different types of maize. Okay. So if you want to start to play around with your breeds and your selections and stuff like that, that's the slide for you. That's it for week 6. There's a few in week 7 for cotton, groundnut, pulse, corn. Yeah, we'll go through them. That's so frustrating when I press open, the double click. Look, open. And it just flashes down the bottom. Please, if you're watching this, can you suggest why that happens? I'd like it just to open. I've got hand problems and using the mouse just, I don't want to click extra stuff. Now I don't know which one was open. There's too many of them. Some seed questions for you. Oh, Etheron, seed dormancy soaking tool. So if you want to break the dormancy, this is a chemical which you can use. So this information in the middle is from Wiki as well, Wikipedia. Ephron is often used on wheat, coffee, tobacco, cotton, and rice to help the plants reach full ripeness more quickly. Because see in the name of it, guess what chemical it is? Oh, it says it there in the middle of the page. Well, anyway, it's ethylene. See it's half spelled ethylene. So that's one of the, remember I said one of the phytohormones that you use in seed germination inside of the seed. It's one of its two it uses, the gibberellic acid and the ethylene. And so therefore, that's why this is a dormancy breaker. So someone just put ethylene into this product, this chemical, and you can have it. Or what I suggest, instead of using ethylene, is use an apple, which is what I use. 1 apple inside of a fruit bag, and then you got your seeds soaking inside containers. Leave it in that. If you actually get because I've done this lots, I do it a fair bit actually, and if you see there's too much ethylene's gone into your germination, you'll get a tripling response, it's called, where the hypocotyl will be thick, so the stem will be short and thick, and that's the 2. And then the 3 is the J hook in the radical. So when the root comes out, that's called the radical, and then it'll put a little J hook in it. And if you'll get those 3 things if you leave it in there too long, and that just means that it's absorbed too much ethylene. So you'll at least know that the fruit or whatever you've got chemical has worked properly. It's a good way to test it. And it's not hurting it. All it does is, because what ethylene does is once it's germinated, it slows the vegetative stage down. So it turns it into a stress, an abiotic stress, and it operates, instead of the growth development pathway, it goes into the stress pathway. So it goes and can regulate things and it can strengthen things. And so it's Not a bad thing to use at all. I wonder if there's any actually questions on this. I'll just stop. I've rambled for a bit. No. Not for now. Advantages of seed hardening. Well, calcium deficiency can be overcome by soaking in calcium chloride solution. About 10 to 15% of the yield can be increased. Seed rate will be reduced. And dead seeds are used for domestic purposes. Seed hardening. Okay. Seed production deficiency. So there's some organic remedies to overcome a few nutrient deficiencies in your seed production. So if you've got light yellow stripes along with veins in the leaf, you've got interveinal chlorosis, and that can be remedied with zinc. Zinc sulfate at 25 kilos per hectare as basal. So you put it on the base, so as a top dress, in other words. That's what that says. Zinc, light yellow stripes, okay. You'd have to know your deficiencies of your areas before going into it, because zinc's very, these are secondary nutrients, which are very particular. So the light yellow stripes along with veins could be nitrogen. So don't go getting the zinc, because it's probably nitrogen if it's that. Iron, boron, sulfur, and calcium. So other ways, spray 1% iron sulfate on 30, 40, and 50 days after sowing for iron chlorosis. Or go and get cytophores. Get microbes with cytophores like, no, I'm not going to say it, so I can't picture it. That's how I'd fix my own problem. Boron, borax, plus gypsum. Borax you can get, it's you can buy those little packets called, I think it's called borax. Sulfur, stunted growth. Yes, thin-stemmed and spindle appearance. Application of gypsum at 45 days after sowing. Oh, yeah, I think sulfur's got the main, very high in gypsum. Well, it's got a good percent in it. Calcium, darkened flower bits, and single-seeded pods. So if you've got a very, not a good application of gypsum to fix it, 45 days. Yeah, if you want more seeds. And to fix more seeds, actually, that's your gibberellic acid. So in flour, it's known to double and triple the seed set. So that's pretty important. So just what it does, swells them. Fair dinkum. Here's some IPM stuff. Really? Okay, IPM stuff must be for people that have seed problems who are producing seed prior to sowing. Prior to sowing, that says. Just sow all over the shop. White grubs, curl grub problems, which you can fix. But rust problems, which you can fix. That's root rot. It can be fixed too. All biological ways you can fix all these problems. For medium-term storage up to a year, the pods can be stored in interwoven polyvinyl bags. Isn't that funny? For short-term storage, seeds can be stored in new gummy bags. Gunny bags. Wow. For seed storage, remember it's in a container, labelled well, in your bag or something labelled well, clip bag, and then put that bag inside a Tupperware container with unexpanded rice and seal it. And then you'll know if you look at it and you've got rice that's expanded, you know you've got problems. You've got to change your Tupperware or your storage thing in the bottom at the back of the fridge. It'll double, triple, quadruple your seed storage time, your seed life, if you've done that from germination. But if you've got seeds from old mate who's been leaving them in his cupboard and it's got up to blooming 100 F, 30, like 40 Celsius, bang, they've nearly almost, good luck. So putting them into the fridge, it's they're already massively dormant. Embryo's probably already dead. So it'll take a good tissue culture section to revive that, to multiply those cells. Seed standards. Oh, here we go. Oh, I wish they knew what the FS and CS means, but this is some standards anyway. Physical purity, you want 96% minimum. Inert matter should be 0. It's 4%. In other words, excessive organic matter. You want none. Other seed, like, but next one, none. Germination kernels, hand shelled. Like if I'm buying seed, I don't want to see stuff in it. So to be a standard, anyway, that's what I reckon. Total weed seeds, none. Yeah, good. Oh, sorry, germination kernels, hand shelled. Hand shelled. This must be for a particular type of something, because you want them all ready to go. Previous container, moisture content, vapor proof. There you go, that's better. See, 9 and 5. Remember before it said 12 and 10 or something? That's a lot better. I support those standards. Here's some ground nut questions. Castor seed, actually castor seed, that's one of the highest MP. If you can get some castor, where you squash it, cake, castor cake, after it's been processed, that's got an NPK of 7, up what double that of chook manure. Oh, the flower of cotton. Wow, isn't it pretty? All fluffy. How to make fertile polyploid hybrids. Well, this is put in the wrong section. Gee whiz. I've done that. That should be in the other section. I'm going to have to go and get... Oh, that's what to put in. How to produce seedless watermelon. Hang on, is this in the... Actually, this is from that class. All right, I'll read it out. How to produce fertile polyploid hybrids. So this is one way of you do it. You take a cutting of, see this branch has 2 ends, so it's got 2 sets of chromosomes, and you want to double them. So you want to make it into a polyploid. So you'll dip some cotton wool. Is this the cotton wool one? No. Well, it is. You get some cotton wool, you apply it to the cotton wool, and then you put it in where the nodes are, right at the base. So you'll cut it off down here and put it down there, or when it's just growing, you'll see one coming out, like maybe up here, and you'll keep it wet. So you wrap it up with plastic, cling wrap, and then that will get into the cells and double it. So after that, you should have a branch with 4N and a branch with 2N. So then you go take your cutting of your 4N, and then you've got a tetraploid species of what you've done that off. Down the bottom is how it happens. The mitotic polyploidy, the formation of 2 tetraploid cells from a diploid mother cell. So it just holds it back at the equator in the metaphase, and therefore it doubles. Basically, instead of halving, it just holds it and it doubles. That's what this that chemical uses. If you're using that chemical, I don't support it. You can use other ones like ORZIN, something like that. But that's poisonous, extremely poisonous. So you have to be very careful using it. This is how they produce seedless watermelon. You can do the same. If you've got a watermelon that you really, really like, You get and do it the same way like they did before. That's one way of doing it on the plant. And then you'll have the tetraploids, and then you'll grab the seeds from the tetraploid, and then you'll go and mix that with the two. So you mix the 4N with the 2N, and then that will make a 3N, which will be the triploid. So that's why, because after you halve a 4N, it goes 2. After you halve a 2N, it goes 1. So it's a triploid way to do it that way. That's how you can do that in any fruit, any plant, anything. Even the 5, if you've got a, I'm not going to go into actually that ploidy. Seed production, dry cracks equal it mature if you're producing cottonseed. A couple of cotton questions. Another dormancy breaker, which could be broken. Pre-soaking. What are they soaking in? Ethereal. See, ETHREL. It's the same thing. It's ethylene. Just another chemical that they put it in, and just use apples. If you really want to learn that more about that, it's called the volatile off-gassing of fruit. So you look up that, then you'll find that there'll be the top 3 are bananas, apples, and pears, and then under that it goes down pretty quickly. So I wouldn't use anything under that. So pick one of those top 3. And then see how they're spraying it with it. You don't have to do that because that's just got the gas in it. So they've got it already in plastic by the look of it. So you just put your apple, just leave it here. Wash it first and maybe disinfect it, the apple, and then just sit it there. And then after you finish with the apple, don't eat it, put it in the fridge. I get about 3 to 4 months out of every apple. And I've done this for many years. And the ripening of the fruit is the off-gassing of the ethylene. So that's, you're going to get it pretty much. And you can therefore also do your tests to see if it's still producing ethylene by the triple response that you get from seed germination. That's it. There's Aussie Jeffro says, "G'day, mate. Hope you're well." Just in the middle of the background. No worries, Aussie Jeffro. Nice to hear from you, mate. Nearly finished this course. You like next week's Aussie Jeffro? It's a microbes in agriculture. Oh yeah, I got my exam results back for those who want to know. See, that's it. I passed everything. That microbes in agriculture class, I got 85%. The food microbiology, I got 82%. And the crop breeding and plant biotechnology, I got 57%. Yes. So here we go. Some questions. Pause them if you want to know questions. And it's been through that. All right, I think it's pretty right. That's the Principles of Seed Technology course. Here's a PDF. Let's have a look at this one. Seed hardness and dormancy breaks. This might be some bonus info because I'm going to put today's title as dormancy breaking. There's nothing open. What do you mean? It's a PDF. A Firefox PDF. Okay, we'll open with Firefox then. Gee whiz. Overcoming legume seed hardness. I can't even put this in the description. Here you go. Actually, there's a few different characteristics of expression of hardness. It's pretty big. I'm going to skim through this, and you can just slow it down if you want to read it. It's pretty big. Very good. The role of the strophile. Okay. Goes in pretty good detail. Breakdown of hardiness. Constant temperatures. Temperature is the whole thing. Remember, 3 things for a seed to break, for it to be favourable for it to actually start to germinate. And it's a good way to think about it with your little ones is picture a baby. Would you put a baby in those conditions? Would you put a baby in hot water? Well, this is a hot water treatment, so it might be different just for rupturing the seed coat. But if you just, put it under, you picture that chemical scarification. Sulfuric acid. Holy mackerel. Oh, just different way. Yeah, sandpapering, dehulling, abrasive. Crack it, just crack it. Remember, it's 3 things. It needs oxygen, temperature, and water. So by doing all this sort of stuff, you knowing the science behind it can make up your own decision on how to open that seed. Is sandpapering really going to work? Or you're just going to get rid of the tiger stripes or the nice looking marks on the outsides of it or something like that? More than likely, it's not going to penetrate. More than likely. But with big, massive seeds like those, like a leopard tree, it probably will work. I used to rub leopard tree seeds on the ground, they get hot. Recent development in Queensland. So there you go. I'll just skim through its last little bit. Conclusion. Each seed lot should be considered individual. Yes, there are ample methods of treating small quantities. Further research is required. Yeah, if you get a lot of seeds that are dormant, you might want to do various techniques to see which one works better with your seed set. You might find ethylene's better, or cracking is better, or a higher temperature, or a lower temperature, or one with gibberellic acid, one with more ethylene. These ways of seed dormancy breaking techniques. Now you have the science, you can put them to test. Wacky do. That's it. Next week, I should see if there's any questions. Actually, I'll see if there's any questions first. Nope. Next week, all excited to introduce. Woohoo! I just finished the course too. I thought this is going to be rad to discuss. What's the name of the course? Is it microbe one? I just got the certificate. That's it, microbial biotechnology. Yeah. Oh, it's week 7. Okay. Microbes in agriculture, biofertilizer, microbial pesticides, integrated pest management. When I was going through the week, I was so excited. I got 100% in this week too. Some questions. What? That's all the slides. So I'm slowly going to go through. There's 5 lectures there for week 7. I'm going to do each lecture as a show, because each slide will be very detailed, and I'm right up on microbes. I've done enough courses now to know it, so I've quite qualified in the area. So I can really go into some good details, suggest some different bios. I'm also doing some other research into, where you live, it gets cold. Most people say, let's pull up a good microbe page. I should just pull up the paper. I'll just pull this up. Everywhere it's cold, they go through winter cycles. So why can't we still grow in that season? So I'm looking into it more, and I thought, because there's a few, there's mesotrophs, which is the temperature range from 15 to 35 C, and then thermophiles, and then to go backwards is the psychrotrophs, which is around freezing level to sort of 10, 15 Celsius, or 50 F, 60 F. Why can't we grow on that? We can, with the right microbes. There's been some studies that have been done on this just recently. And I thought, you beauty. I just love staying up. I love having the science behind it, so I can put the science into the experiments to see how we can better get better plants, better, healthier production, bring out the genetic potential, grow in off seasons, produce perennials when they're only annuals. Weird stuff in science, plant science is what I love to do. That'd be a good title, Weird Stuff in Plant Science, because that's what I like to do. Like I've got some, anyway, that's what's coming up. So if you're into that, tune in. It should be great fun. So I really enjoy it, especially the microbes. Oh, I still got to speak to, yes. Anyway, put some words into the chat if you want now, because we're going to be wrapping the show up soon. And I'll hope that next week you can bring all your microbe questions and we can get stuck into it. And then I'll go through that psychro-tolerant microbes as well, because it is to do with agriculture. So I might make a special week, maybe last week or something, and talk about new technologies in microbes and agriculture, which that is involved in. Because the paper, where is it? Oh, here it is here. What year was it written? I only just start reading it. And of course, it's from India. Yeah, 2021, late to November. So it's only a few years old. We'll go down to where I was up to. So awesome. Yeah, I've got to remember I'm doing a live show here. The microbes, they rule everything. They are everything. And now science is getting into it more with our microscopes. They're finding that it rules humans. 90% of our human cells are microbe cells. So it's just excellent. And then getting the right species. Like here you go. If you live in, I should be talking about this next week. For those who are still listening, which there's a few of yous, if you live in a cold region, this is what I want to do. I want to go to up on top of the mountains, maybe there might be glaciers around, and pull some plants. And where their roots are, I want to get there and sort of nearly even cut the plants off at the top, the phytosphere bit, the green bits, and then keep the roots down to sort of 5 centimeters, down to 2 inches, and the soil as well. And bring it back and do some spread plate cultures. So get some of that material and put it through various different cultures under different media, and you'll probably find that you've got some microbes that are new to science, because people think nothing used to grow in those conditions. Well, they were wrong, because the protozoa proved them wrong with all the extremophiles. But those ones around the ice, because I was racking my brain, I thought that's the go. You can get your garden to work in winter, or your agricultural field can be thoroughly green, producing your crop in winter, where normally it cycles very, very low. You can get nitrogen cyclers that, instead of doing 10% at, say, 10 Celsius or 50 F, they might be doing 30 or 40% nutrient cycling of nitrogen. So that's going to boost the plant in that sense. You get other microbes working for you. So from the psychrotrophs, you can, if going and getting those types and bringing them into your region, actually, there's biosecurity problems and all sorts. So probably don't do it. But that's something that interests me anyway, and I'd be doing it under strict conditions and all sorts. But yeah, because there is, but it is, you don't know what those microbes are. They could have been the plague that you're unlocking, because it's very, very cold. So you've got to be pretty careful too. Hey, there's Lene. Geez, all these things. He's just waking up, listening as I down a couple of minutes away. You just missed the last week of the seed production technology class, which was all right. And it was also on seed dormancy. That's what it was. Yeah. And I'm just talking about now getting, well, I can ramble for about microbes, because the more I learn about them, the more I find out just, wow, why aren't we doing more of this? Why aren't, and they're kind of cheap as well, from what I've found, if you buy in large amounts. A lot of manufacturers and a lot of people now, they've gotten onto it, these scientific papers, and they think, oh, cool, I can go and use Lactobacillus bifidum as a probiotic. So they'll just get that one microbe, put it in a capsule with all sorts of other, with their bulking agent. So it'll have 0.01% of that microbe, and they'll charge you $1 a tablet. And it's just really eye-opening for me. There's huge money to be made over something that's so, well, mega cheap to start with. You can get this stuff. Oh, I'm not going to tell you, actually. Hey, Sunshine says, g'day. Oh, yeah. Lene says, g'day. That's the way. I might be rambling now, but yeah, if you've got any seed questions, put them in, because this is the last week of the seed bit. If you've got anything on maybe dormancy breaking or seed soak solutions, remember that nanotechnology and agriculture class went through quite a lot of seed soak suggestions where they've got NPK. Actually, that's very relevant. They've got NPK alternatives. So don't go, I'm all about organics. Don't buy fertilizer. Make it. Microbe it. Where is it? That class. This one, nanotechnology and agriculture. Here we go. There's a lot of seed stuff in here too. So I'm not going to go through all the slides. This would be actually, this is one I should be doing too. Oh, all it does is flash down the bottom. If you're in chat, why does it do that? Why, when I do this, press open, why won't it just go boop, boop? Or how can I fix that? It's just doing that. Just does it to mess with me, because it wants to wear my hands out. My hands are already sore as to start with. So this is the seed soak. So the seed pre-treatment. So you might soak it with your different elements, and they're finding that when you're growing it, because of the seed element that it's soaked in, it's chemically produced an NPK pathway for the plant. And for somehow, all the plants that have been tested, and they've tested about 20 different varieties of vegetables, they're all producing copious amounts higher than the ones in control. So by soaking them it really, really helps. Iron pyrite is a really good one. Iron pyrite, it acts as amylase. Where is it written? It's not. Well, that's what it does anyway. And amylase is one of the enzymes in the seed that the seed produces to break down the starch to get its energy. So we're putting iron pyrite down. That's one of the nanomaterials they're finding that helps massively in seed germination. Let's just go on, but it's the same one. Metal sulfides, that's iron sulfite doing the same thing. He's very smart, this fella. There was no text in the whole of the course. He just did it all off his top of his head and drew things like the bilipid bilayer up top, all sorts of stuff. And he just explained it. I'm just spinning out. Some other professors, they just read. This fella, he drew, and I was just, whoa. Quite interesting. But back to the seeds. Ion particles, different ways to do it. Stoichiometric effects where it swings up or down, goes from a 2, FeO2, from a ferric to a ferrous. See, they soak chickpea and spinach and rice. Chickpeas are finding that the high germination percents on the bottom right, the root growth was higher, shoot growth higher, overall biomass higher, just from soaking the seeds in iron pyrite. Which are seeds treated with iron pyrite has higher ability to harvest sunlight as well. Can you believe that? Because it puts out a bigger leaf size, which has more cells, which can obtain more chlorophyll and produce more energy. So you're going to get more biomass, and that's how that works. Is this another one? That's still talking about the same thing, just different field of trials, how it works, which just works. Now it's talking about all the different types of ones that Sis has done at the University, IIT, Kanpur Uni Nursery, talking about the ones that I've done there. Now, all of this improves everything. High chlorophyll, high magnesium, high sulfur content, high calcium content. Can you even believe? So it's new technology. It's nanotechnology in agriculture. Catalase, superoxide, dismutase. Oh, that's the reactive oxygen species. So it also lowers stress by putting out antioxidant enzymes. How cool. Nanopyrite down the bottom. NPK equivalent in rice production. Here's the yields. 1.3 there with NPK, 1.6 with the iron pyrite, minus NPK, 1.63, iron pyrite plus NPK, 1.68 as you'd expect. So look at that, NPK or iron pyrite. And now all of this is talk about carrot, fenugreek, mustard, alfalfa. All the same thing has been done here with treatments of not just iron pyrite, calcium, manganese, zinc, sulfur. So different separate ones with separate elements. So if you've got a chance to get separate elements, soak your seeds in them. Write down the mixes and the ppm, the amount you put in, and document it. That will be interesting. And they're fine. Instead of 4.6 kilo in beetroot, 8.6, 47% increase, 19% in carrots, 65% in mustard. And further, iron pyrite water system is an artificial starch breaking enzyme. Oh, that's what I spoke about before. The amylase, there you go, see? Just like amylase, it breaks down or chops up the glycosified linkages, therefore increasing the amount of energy that can be made. Look at that, all these, this does to this, and this flows to this, and this flows to that. It's so excellent. On this, I've already chosen my subjects for next semester. So they go and sulk with AG. Isn't that, is that silver or gold? Well, anyway, AG as well. Carbon nano onions, carbon nanotubes. So it's the new, beyond the NPK. So this is the new organic thing, because the farming of the NPK supplements, is it the phosphorus, rock mining, I think? Was it my professor? Someone was telling me that it's, or was it the neighbour? Someone was telling me that they can't produce as much as they used to, and there's going to be a shortage coming up. Finally, the world's used too much. So they've got to do other organic practices soon. So from 2030 and 2040, definitely, this will be all second nature to everybody. We'll all be talking about organic this and how to do that, because that's what the world will, the market, and everything's about marketing, because everybody's already been sold NPK is the best. That's false. Nanomaterial, yeah, you can even, on it, I'm talking about seeds, focus here. Yeah, nanomaterials are helping in reducing NPK requirements. We spoke about that. Nanomaterials, okay. To get the nanomaterial across, you've got to have a vector of some type, and these are the different vectors. Oh, there's the list. Even citric acid. Look at that. You've got, that's the seed soak list. So if you want to try some new age, next decade technology, start practicing. You'll be right up there with the best, because you'll have, I've done experiments, and there's nothing like speaking from experience, because it's just happened. And if you can remember it, that's why writing it down helps, because you can remember it in your brain, but then by writing it down, because I write my stuff on my calendar or on bits of A4, used A4, I turn over, keep in a pile. And it helps verify, because you said it once and you're already discussing it in your brain, and then writing it, you sort of discussed it twice. So it just, it's rote learned the information into you. So it helps also to, when you want to say it later on, yeah, I did a seed soak with iron pyrite and I got these results, or I got that, because you can picture what you've written down. That's how it helps me. All right. An hour. See how microbes, they're excellent. Nanotechnology, it's good. Oh, GMF, no. GMF says, good morning, good peeps. I hope you all are safe and happy. Good evening to the goats as well. You just missed it, mate. Watch the last 20 minutes of today's show. I mean, you can watch the whole thing, but the last 20 minutes especially, you'll really enjoy, because I went over some seed dormancy tools, seed breaking, and went into nanotechnology, how to use that to your advantage with seeds. And talked a little bit about microbes. Our next week is going to be microbes in agriculture. And I'm not starting again on microbes, because I talked a lot on it. So that's it. I was just signing off. So thanks, saying good day. It's GMF. I hope you're well, mate. You have to get onto Zoom again, GMF. I'm going to go. And I enjoyed today. Thanks, everybody, for writing stuff and listening, giving me your attention and re-listening this back if you are. Press the like button if you liked it. And next week, we can get into the microbes again. Woohoo! It's going to be good. GMF said, just woke up from a nap. Sorry. Thanks for the show, Aussie. You're welcome, mate. It's welcome. Yeah, press the like if you liked it. I keep forgetting to say that, and I've said it 3 times in this show. It's a bit annoying. It seems like I'm hunting for likes. I'm not really hunting for likes. I'm hunting for, I don't know, more ears, more communication, more interaction. Yeah, I enjoy helping people. So to get specific questions is I really enjoy too. But it's nice to also catch up with the cool folk in the community. Like you can see here, like GMF, Lene, Sunshine RB, Aussie Jeffro. Sure there's other people saying thanks to subscribers. I'm not sure if they want to be mentioned, but I really appreciate that. I see that was just redone a few days ago, so thank you. Well, GMF says, chat, leave a comment after the show, helps the algorithm. Oh, okay. Oh, isn't it more than 7 words or something? Because under that, it doesn't really mean much. That was something I think you taught me a few shows ago about that. So I started to look at that because, yeah, I don't get anything out of this show. No real money, no nothing. So those things help me a lot. The words, the comments that people say, and the likes, and yeah, other interactions. I appreciate everybody who tries to help, because I think a few years to go with doing this stream, because I got a few years to go in my studies, and my next semester I've got environmental science, cell signaling, applied entomology, and that's going to be good in specific applied entomology for the insects that are important for agriculture. I was very curious about that. Some IPM, some pest management. They go into the bios, beneficial insects. So I've really thought that those sections, that's why I'm doing that course, because that can benefit me from doing that one. I've got an environmental food and dairy microbiology. I'm not sure, because I just finished a food microbiology one, if I should. And there's plant physiology and plant tissue culture I might do as well. I've already done that from a different uni, but it's offered. I like doing it because sometimes they will say different things that other universities didn't mention, and I can always pick up on that, and that means that I'll learn one or two things out of that, and I can share that with you. Holy mackerel! There's Teo. Hey, thanks, Teo. Look at that American one. Nice to see you, mate. He sent $10. Jesus, just appreciate it. Good on you, mate. It's just talking about microbes. You're into your microbes. Next week onwards is a microbe. I go into the microbes in agriculture, so it should be pretty cool. And I'm doing some new how you can use your garden in winter research, which is fascinating. And it's in new technologies. But I'll go through new technologies in agricultural microbes probably in the last week. It's rad. So that's what's happening. Don't miss those ones. The American one, you like that? I remember you're into your microbes. It says, g'day. Hello. G'day, Aussie CC. G'day, Genetic Memory Farms. Hello, Lene. Taylor says, I appreciate the effort and knowledge. Yeah, thanks, mate. And I like the nice words and effort that you and everyone else puts in too. Appreciate that. It's really good. I said to GMF before, I have to get back on Zoom again. Have some fun, like talking agricultural stuff. Yes. Yeah, and I got 85% in that course too, the microbes in agricultural class from my exam. I got the certificate already, so it's pretty cool. I was quite high. Out of the 1,400 people that did the class at university last semester. I love the study. It's so fun because you get I don't know, it's just you learn what the information is like from you knowing now about the 3 things that a seed needs. You can go and suggest all these dormancy breaking tools and techniques to achieve that. So people says, oh, you have to use sandpaper. Not just sandpaper. Well, you know why you can use sandpaper and why I might use pliers, or I might just use a knife. These types of things. So by knowing the science behind the processes, you can change things and tweak little situations to work to your advantage. That's the bit that I like. Yeah. What? Has he gone and done another $10? What's this with inflation and tariffs? $10 doesn't go far. Does that mean he did another 10, mate? Looks sort of like it. Holy mackerel. Making it rain, freedom notes. There you go. Might have to stay on and talk a bit longer at this rate. I was going to sign off 10 minutes ago, just when GMF come on. And this is why I've stayed these extra 11 minutes of Holy mackerel. $20 off 11 minutes. What's that? Times 6. Gee whiz, that's a good hourly rate. Thank you again. And it's the words and efforts like this that make me get excited and keep going to share the information. Keep me going in my studies, because I know, all right, cool, this would be good for everyone to know. Oh, this is relevant in our gardening, horticultural, agricultural sector. Oh, this is an organic task or an organic process that we can introduce that's going to get better results, like the nanotechnology in agriculture, for instance. All this stuff, it's I love it. GMF says, the American one making it rain. Oh, yeah. American one says, and yes, I believe in microbes. The worms are my best friends. The worms. Did you know the outside of the worms, they secrete ammonium? One of my teachers was a biology professor on worms. She loved it. It was her worms, and she went into a bit extra detail on the worms, the 3 different types of worms that we have in our gardens. And the epizoic, anazoic, and endozoic, and that the big massive ones that you want for fishing, they eat worm castings, so look out for them. Worms rule. They do so. They vector our microbes around and fungus spores, because bacteria has a lifespan of a dot on a pencil and paper, so that's the amount that it can travel. So we need these types of things to get around, let alone secreting ammonium from its skin. How cool is that? Putting out the castings, which are so rich in the moisture holding capacity. What's that called? So moisture retention? No. But yes, worms are good. That's in the, I went through, did I go through the organic farming? I think that was in the class that she was helping to teach. There's a lot of worm stuff in that about the 3 different types. Actually, I just pulled out a big, massive one the other day, and I thought, I'm not putting this back into the veggie garden because it was too long. Of the 3 different types that there are, because the normal ones we want is just, they're going to produce the worm castings from the organic matter, but the big ones, they're going to eat the worm castings. And they're the ones that are longer than about, what's that, 6 inches, 125 mil, I'd say 150 mil. So if they're longer than that, I wouldn't be putting them back in my garden, because they're more than likely, they're of the species that eats worm castings. So, yeah, just something I thought I'd top out, because you said you're into worms. It's come off the top of my head. Worm head. And if you aren't wanting to go into detail, ask me on another show, Teo, or even now, and I can elaborate if you want me to look into the difference. Because you want to, I got the different species names and genuses of the composting worms that are more beneficial. Ones eat more than others, and ones cycle at different temperatures. So you might want ones, you're in a colder region, so you want ones that are the colder varieties, for instance. Or you could get the ones that you've already got, then the species that are the cold ones, and then put them in favourable conditions for them to lay eggs and keep that producing. Yeah, this organic stuff's rad. It's the future. Remember that the NPKs aren't being produced as well as they used to. They're starting to run out of the phosphorus from the rocks. So things are going to change in the decades to come. Everyone will be going back to organics the traditional way, because that's the way how it's been up until last modern era, 200 years ago. Man ruining the planet. Bioremediation, bio-augmentation. I'm rambling. Worms rule. Okay. I should go. I'm all excited now because you got me excited. Yes, you. Teo says, yeah, some go deeper, some stay on the surface more. I have a worm bin. I have red wigglers. I don't know the scientific name, but they are happy and make plants happy. Yes, they're the ones that endozoic or epizoic ones that are on the top that eat the organic matter, the red wigglers. They're not the aneic, ANEIC type of worms. Is it the family, the class or order? I'm not sure. It's quite high up anyway, but they're not the ones in the red wigglers. They're rad, which makes me happy. Yeah. Worms are good. I don't know what else I could say with the worms. Yeah, there is a few different types of composting worms that do a better job. And the composting things. What's those things that I keep throwing in? The curl grubs. Oh no, we're talking about worms. I should stay on point. Yeah, the only thing with worms is if you like chooks, your chooks like worms too. Because I got chooks. So I only let them out at certain times now, because if they come out, they'll be scratching and eating all the earthworms first before anything else. No, I want them to start reproducing. I need them. Well, I'm going to be rambling, so I think I should sign off. Start asking questions or something or other. Otherwise, I'm going to sign off, I think, because I don't like to hold people up, if you know what I mean. I don't like to be, let's learn something. Let's interact. Let's make it interesting. Yes. So that's why I can, what's Lene say? Excellent show as usual. Thank you, Lene. Enjoy your weekend. Good on you, Lene. Thanks for your nice words. Yes, I hope so. I look forward to next week with microbes in agriculture. And thank you for the support again from the American one. I hope to see you and others in the next stream, which starts about an hour, 20 minutes before this one next week. So whatever time zone you're in, it's 11 a.m. where I am. But you got to work that out, because time zones change all the time with the blooming daylight saving stuff. 1 week it's 2 weeks ahead, 1 week's, because mine will go back, yours will go forward, and all of a sudden, oh, gee, 2 weeks apart. And anyway, thanks again for your ears. And I hope you got something out of the stream. And you gave me something too. Thank you. So happy breeding, happy growing, and good health to you all. Hope you have a great week and week, everyone. I hope to see those on Zoom. Good night. Good day to you. Bye-bye.